Speaker of session 2 is Duxin Sun, and the title of his talk is Broccoli as Functional Food to Eliminate Cancer Stem Cells. And he's from the Department of Pharmaceutical Sciences.
Thank you. My talk literally is standing right between you and lunch, but I promise you, give you some advice for you how to eat broccoli at lunch if they provide broccoli. Most likely they did it wrong. So WHO issued a report a few years ago. Literally in that report, they say cancer is going to be a human disaster in the next 20 years. There are going to be 14 to 22 million diagnoses every year worldwide. So if you look around you, beside you, statistically speaking, one of you will diagnosed with cancer in their lifetime. So that's actually pretty scary. And the situation getting worse, not better. So I think as a cancer researcher, we perhaps did a poor job as compared to other disease areas like cardiovascular, infectious disease. So for breast cancer, I'll take this breast cancer example. Breast cancer, you have a 4 types of breast cancer, luminal A, luminal B. They are characterized by the ER, PR, and those 2 receptors. If you have those type of breast cancer, it's relatively easy to treat. Tamoxifen is the typical standard treatment option. The third category is HER2 positive. Breast cancer expresses a HER2 oncogene. And then in the last 20 years, there's an antibody called Herceptin, works really well. The problem is after 1 year treatment, those cancers develop a resistant. So once you have resistant, there's no other treatment option. The worst case is the triple negative. If somebody have triple negative, it's called basal-like. You have ER negative, PR negative, HER2 negative. So estrogen receptor, proestrogen receptor, and HER2 negative. Those are the problem is there's no treatment option. So the best option right now is chemotherapy. It's very toxic. They have a much higher metastatic potential. The recurrence rate is higher. Then it have a poor survival. So now, the reason in cancer research does not have much improvement as compared to other areas because perhaps many of the discovery or drug treatment we have does not work well. So you need a new theory. So about 5 to 10 years ago, one new theory came out is called cancer stem cell theory. It's more like an embryonic stem cell. In cancer tissue, you have a very small population of cancer stem cell. They will differentiate it to cancer cell. Then only cancer stem cell can form tumor and initiate the tumor to migrate to other tissues to have a metastasis. And those differentiated cancer cells is relatively easy to kill. They will die gradually. However, you do need to treat them. Otherwise, the size of the cancer will kill the patient. Versus traditional model is every cancer cell is able to form tumor. Therefore, you need to kill all of them. So those are 2 different models. Perhaps we don't know which one is true, which one is not true. There's a lot of debate about the cancer model. However, one thing for sure is regardless you believe or don't believe cancer stem cell model, if you use a traditional chemotherapy, you perhaps will kill most sensitive cancer cell. Then you have a drug resistance. If you believe cancer stem cell will stay behind. Then after 5 years later, you remove the drug, they will reoccur in the same site. They will move to liver, to bone, to brain, form a metastasis site. So ideally, then you want to find a chemotherapy drug or drug can kill both differentiated cancer cell and cancer stem cell. So the problem right now is most chemotherapy drugs, they only able to inhibit differentiated cancer cell. They are incapable to kill cancer stem cell. So therefore, if this is the case, the cancer will never cure. So they will always come back. So now let's come to a broccoli as a dietary component, right? The question is, number 1, I want to give you some of the quick data is, do dietary component have anti-cancer efficacy? For broccoli, there are 20 years literature saying, oh, broccoli is a wonderful thing to can kill cancer cell, have anti-cancer efficacy. if you have epidemiology study, the region who consume more broccoli, they have a lower incidence of cancer incidence. Now, is that a coincidence? Is that true? If you use a broccoli extract or the active compound to do clinical trial, it's very controversial. There's no clear efficacy in clinical study. So whether it's true or not is hard to say. That's number one. Number two, then how about the cancer stem cell? Because we feel some of the natural products, some of the dietary compound, oh, you have a lot of story, heard somebody eat this, they cure the cancer. Is that true? It's just failing? And then whether they will have effect inhibit cancer stem cell. Number three, if they do, what is the real molecular target? I think there's a lot of voodoo science into this area. Who knows it's true or not true? It's a lot of speculation and not solid data. So then if it's true, we need to provide a solid data to see it's true or not true. And then number four is how do you use it? How much you need to use? So when we publish this data, they will ask then how much I should eat. Should you eat 50 kilogram per day, or should I eat 100 gram a day? So broccoli is very well studied for 20 years. The active compound called sulforaphane. Broccoli sprout has more of those than broccoli. They inhibit multiple targets. Now, the first question is, do they show anti-cancer efficacy? In vitro, if it's a HER2 positive breast cancer, no, they don't. You can eat 50 kilograms, they have zero effect. If you have a triple luminal A, luminal B, it's a little bit better. But if it's a triple negative, you have a much better efficacy. So the SC50 around 7 to 10 micromolar. 7 to 10 micromolar is not a very good drug. If it's drug, it has to be nanomolar range. But this is the food. If it's a nanomolar range, then everybody will die, right? So depends what type of cancer you're talking about. Then if it's a HER2 positive breast cancer, you can eat tons of that. You can have 100 or 1,000 micromolar concentration. They don't do anything. However, if they develop a drug resistance, especially for patients with the PTEN deletion, which is found in 40% breast cancer, then at that moment, then sulforaphane does work well, around 5 to 10 micromolar. So you do see this very big difference. So which is a good thing, perhaps, for this. Now, how about stem cell? Can they eliminate the cancer stem cell? There's two types of stem cell in breast cancer. One is MET-like. They are more proliferative. We use ALDH as a marker. And another type is called CD44 positive, CD24 negative. They are more quiescent. So by flow, you can identify the stem cell. If it's a control cell, it's a very small population of stem cell, about 3 to 5% in patient tissue, cancer tissue. Now, if you have a 1 micromolar or 5 micromolar sulforaphane, you can significantly reduce the stem cell number. This is just a number. So those are the movie I show you. We can track the cancer stem cell by CRISPR. So the red are breast cancer stem cell, the red cell. On the top is the control, no drug treatment. You can see the red, they are growing, they are proliferating. On the bottom is under the sulforaphane, 5 to 10 micromolar. You can still have a red cell. At the end of the movie, you see less red cell. At the same time, you can see some of the red cells, they try to divide, they cannot, they stopped. Of course, there's no way you can eliminate 100% of those. So that's in vitro. How about in vivo? Right? Then it's very controversial to use a dietary component to evaluate anti-cancer efficacy in vivo. So if it's in vivo, most we do, we implant the cancer, then after a while, we measure the volume. That's traditional way to measure it. Dietary component is actually not bad. However, in terms of anti-cancer efficacy, if a drug only inhibits 40% of cancer growth, we consider that inactive. It's very hard for broccoli to inhibit 40% cancer growth. 40% cancer growth, you still have cancer. You still will die of it. So, however, this cannot tell the stem cell status. If you take the same amount of cell from here and versus here, that's the volume, that's treatment, then implant to secondary mice, then if there's no stem cell here, they will not grow. If there's stem cell here, they will grow. By definition, only stem cell can form tumor. So this is the log scale. So this is the log scale. You have very little tumor growth and a very large tumor growth until this day. Of course, there's not 100%. After a while, they will still come back. So sulforaphane would be not that effective. So, didn't show. So then it does show, and also the concentration for inhibited cancer stem cell, it seems lower than the concentration inhibitor differentiated cancer cell. Seems the sulforaphane preferentially inhibit cancer stem cell. So then the question is, what's the molecular target? I didn't show the heat map here. We did RNA-seq analysis to see then in the sensitive and resistant cancer cell to sulforaphane, what are the molecular target? By identify this 144 gene target, by knocking down 40 of those, we identify quite a few. Actually, after that, we found different type of cancer, breast cancer, they have different target by sulforaphane. If it's in triple-negative breast cancer, they actually influence the interleukin-6 alpha-kappa B loop because this is actually regulated triple-negative breast cancer stem cell. So they reduce the interleukin-6 and 8. They also block the translocation of NF-kappa B2 nucleus. Therefore, they cannot transcribe all the downstream target genes. That's in triple-negative breast cancer. If it's in HER2 positive, drug resistance, when you have a PTEN deletion, the target is the MUX1. So the MUX1 is a transcriptional factor. They actually inhibited this, and this target is actually correlated with the patient survival in the PTEN deletion P53 now human patient. So really, then the same dietary component in different cancers, they have different efficacy or they have a different molecular target. So we published this data, then it went to the news because it's broccoli, because it's breast cancer, because President Bush does not like broccoli. So it all went to the news. Then we start to receive an email say, hey, then how much I should eat? I said, I have no idea. So we have no idea. You need to eat a tenth of those, then you never be able to achieve it. Then we'll say, let's study it. So my student did it. In broccoli, it's very interesting. So the precursor of sulforaphane is glaucoraphanes. They have a sugar here. Then one enzyme called myrosinase will cleave here. They have an intermediate. Then in broccoli, there's another enzyme called ESP, or convert this one to here is sulforaphane nitrile. 90% of the come to this way is toxic inactive. Only 15% going this way. So in broccoli, all those are in different compartment. If you eat fresh during the process progress, everything will mix together. Most of them are going to here. Therefore, if you eat fresh broccoli, it's useless. So that's different from the other nutritional study. So if you do fresh or you steam, you kill the enzyme. Then we'll stuck everything on the precursor. Or you steam, you kill the enzyme, you add one enzyme on the top, you don't add another enzyme, they will convert all the way to sulforaphane. So by doing that, you are able to achieve enough concentration. So fresh, nothing there. So precursor, sulforaphane, all gone. Steamed, only precursor. If you do really correctly, you have a more sulforaphane. And if you measure the concentration in blood, in breast, in all the tissues, only at that time, you are able to reach minimum effective concentration. So really depends how do you cook it, how do you eat it. So it could be good or bad. So to quickly summarize before your lunch, I think I'm on time, is dietary sulforaphane broccoli, they actually works only in triple-negative breast cancer and only works on drug resistance, PTEN deletion, HER2 positive breast cancer. So other two types don't work. They actually preferentially eliminate breast cancer stem cell, which much lower concentration, to 20 times lower than regular cancer cell. So they have a different target in different cancer, different type of cancer. Only if you correctly cook it, you eat, it's about 100 grams, you may able to reach enough concentration. If you don't cook correctly, perhaps you can eat a kilogram, this may not reach the concentration. So really, that's the estimate we have for how much you should eat. So that's my lab, and a few students did, my former student here, and Joe in this team. We actually have a cancer stem cell natural product team. So I don't have time to talk about other nano and drug discovery, drug optimization team. So all the credit go to them. Thank you very much. I am on time before your lunch.
Does this sulforaphane occur in, say, kale or other family in the cabbage family, other plants in the cabbage family?
Yes, it's actually in the what are the name
Crucifery.
of Crucifery, the species. They have the broccoli, flower, all the spicy stuff should have this. Sulforaphane is one of the compounds. You have a similar analogs. They perhaps have a little bit higher or low activity. So those family of the plant do have those class of compound.
Does the mustard have a high concentration of this compound?
I don't know how it would be compared to mustard. For sure they have it, but I did not compare which one is higher. So my assumption is that the spiciness of mustard is because of one of those, but I have no clue is that the only thing cause the spiciness or others. But I doubt. There might be something else.
Thank you.
Hi.
Yes.
Thank you for your talk. I'm curious how you determined, or how prior research determined, that sulforaphane was, I guess, in isolation effective in its anti-cancer treatment versus a more, like an extract that includes other plant chemicals that could be interacting, and why you chose to use sulforaphane alone in the experiment, and if you extracted it from actual broccoli, or if you know of studies that have looked at more holistic analyses of different chemical compounds.
Yes. This one is from the processed food. Okay. And the earlier data we have is a pure compound. So there's few issues. If you have an extraction, then you have a mixture of other active components. This may be better. Then the problem is, how do you control the quality, make sure they are there? So that's number one. Two is, sulforaphane is not very stable, right? When you eat it, and this actually processed the food, we gouge the mice. We know they can be absorbed. But then also another speaker earlier said how much drug are going to be absorbed, and bioavailability is how much. That's also a factor. So right now, you can buy sulforaphane extract from CVS. It's all different kind. We don't know which kind is better because I'm sure the content is different, the quality control is different, and there's no regulation.
Thank you.
Yep, there.
Thank you for your talk. Can we prepare the sulforaphane in the lab, like in active form? This is one question, to give it to the cancer patient. And the other question was, like, when you give it to the patient, was it with the chemotherapy or without chemotherapy?
So we don't think if you only use dietary, this one will cure cancer. That's not possible. We did publish, if you combine with the paclitaxel, so paclitaxel is first-line therapy for triple-negative breast cancer, then when you have to use paclitaxel to kill them, otherwise the patient will die with cancer. However, paclitaxel do not kill cancer stem cell. They actually enrich cancer stem cell population in the residual tumor. That has been confirmed by many labs. So that's the side effect or the problem when you only use paclitaxel. We did publish data if you combine sulforaphane and paclitaxel together. Indeed, in mice, they're doing much better. Clinically, no, because this is not approved drug. FDA is not approving this in the human trial. So whoever want to use it, you can use your own by the food supplement. And that's where actually we get a lot of email from the patient to see, tell me how much we eat, how much we should eat. I often very cautious because I just send them paper. And because I said, number one, I'm not a physician. I cannot give you direct advice. Number two, we actually did not test in clinical trial. So ideally, you test that in clinical trial.
But ethically, can we recommend it to the patient to take it with the chemotherapy, or?
Do I advise?
Yeah.
Yes, I would advise.
So just as supplement, not a complementary treatment or supplement, not as a
supplement.
Treatment.
Yes.
Okay.
So then, because the supplements are already available in the store.
Thanks.
So sorry, I can't name the different compound.
Sulforaphane.
Go back to the slide with the, yeah. So you say this is 85 to 90% toxic. Is there any study trying to stop this metabolic step to, in the plant, so that they were direct? Is there any this kind of
I
study?
never heard. I actually tried to talk to, because I'm not in your field. I'm in pharmacy field, right? What if somebody say, I can use not GMO? No, how about you can breed a broccoli, you breed this out. You breed this out, you only keep this enzyme. Then you can only go this way. Genetically, it's very easy to make it. You can delete that enzyme, right? So that's way to go. I never heard it. I never heard about this. So I promise to give you a recipe. The best recipe to eat broccoli is steam first, kill this, leave everything here, then mix with the Japanese daikon. Japanese daikon only have this, they don't have this. So that's the best recipe.
Japanese what?
Daikon. The radish.
Okay.
So I go to Jimmy John's, I always order the Brussels sprout, the freebies. I know it's fresh, but give me a comfortable.
Okay. Well, thank you.
There's a last one. Oh, there's a lot of reasons. One is that their signaling is different. Signaling pathway are different. Second is they don't divide. They're very quiescent. This just all the regular. They want to survive, right? They don't want to divide. So they only maintain 3 to 5% population in the tumor. So if you isolate 100% stem cell you culture, then after you get 100 cells, 5 will be stem cell, 95 will be non-stem cell. So it's very similar to embryonic stem cell. That's the property of the stem cell.
Let's thank Duxin and
Thank
all
you.
the speakers for session 2. It was great talks.
Now it's lunchtime in the hallway, so we will reconvene at 1:30 for the afternoon session. Thank you very much. That was a really interesting talk.
Thank you.
Yeah.
Thank you.
I think we badly